from Microbe TV. This is Office Hours for Wednesday, August 2025. I'm Vincent Racaniello, and welcome to your viral corner of the internet, where you can ask questions about viruses and other things too, sciencey things, life sciencey things. Yeah, we even talk about outer space, but we're experts on viruses here, and you can ask me anything. Thanks for coming, everyone, tonight. Rainy evening here on the East Coast. Chilly. Don't need the AC anymore, that's for sure. Let's see what we have here. 19°C and raining. Look at that. What are we going to have tomorrow? Raining also. Friday, Saturday, it's going to clear up. Is it going to get warm again? You know, it always does in September, October. Thanks to our mods for coming tonight. We have Les and Tom and Vanity Nutrition. Thank you for coming and keeping things civilized here. What's on the plate for tonight? Well, we have all of you is on the plate. We have a little bit of news, one news item, and I have a mini lecture on nucleic acid amplification. And by the way, Dan Wilson, has agreed to come on Office Hours. He's been on here before. He reminded me, and I forgot. But it's a long time ago. He'll figure out a Wednesday that works for him. Anybody else you want, let me know. We see if we can get them. You know, you need to be a certain kind to really do well here on Office Hours. You have to feel comfortable with uncertainty and questions coming out of nowhere, you know, that sort of thing. Let's see where you're all from. Tell us where you are, and we will shout you out. Lise is here from Columbus. 22.7°C. EHS Talent is in Pennsylvania. Fields of Green is from the Salinas Valley in California. 76F and sunny. So happy to finally be free on a Wednesday night to view the live stream. I think I saw you on Tweevo this afternoon, right? Welcome back, and glad you could make it. 76F. I would like 76F outside. I don't like cool weather. I want it to be warm. Virus's Luna is in Iowa, 25 to 27°C. Virus's Luna wants an episode about cat viruses. You want a TWIV, or do you want a live stream? You want? I don't know if one person can talk about all cat viruses. You know, they would talk about it in different things. I could do a mini lecture on cat viruses. I'm capable. I don't know the veterinary stuff, but I'm capable. Hello, Will from Changsha, 30°C and cloudy. Mike and Lori are in Burien, Washington, 23 and sunny. Welcome. Lori is here, says, please hit the thumbs up. We know this will be outstanding. Once a guy was here, he said, I'm not going to give it a thumbs up at the beginning. I don't know if it's going to be any good. Have you ever had a bad Office Hours where you didn't learn anything? I don't feel there's been a bad one. I feel that everyone is always engaged in asking questions, and I love them all, each and every one of them. So hit the like. We got 81 people. 81 people and 41 likes. 2-to-1 ratio. Let's do it, folks. Let's do it. John wanted to know if cat viruses have 9 lives. Now, if you define a virus as an organism with 2 phases, a living, 2, not 3, the third finger wants to come up, a living and a non-living phase, then you could talk about 9 lives. That's okay. But if you're talking about the virus particle, that's the non-living part, right? Elizabeth is in West Virginia, where it's 21C. Kang is in Chicago. 21C, cloudy skies. Thanks for sharing Frank Zappa's quote. A mind is like a parachute. It doesn't work if it's not open, if for keeping my mind open. I hope so. I try to keep my mind open. I'm open to things that I may think work one way, and then I learn that they work another. You have to have an open mind, not just as a scientist, as an inhabitant of planet Earth, as a human being. You need to have an open mind. That's where the world gets in trouble. People don't have open minds. The current situation in the U.S. is because a bunch of people have closed minds about who should be running this country and in what way they should be doing it. That's a closed mind. It doesn't work. We'll see what the outcome is. Hopefully people will come to their senses. John is in Minneapolis, 26C. Kip, San Francisco. Hello, Kip. How are you? 22C. Rita is in Idaho. I know that. See, I'm getting to know people just from their names, where they are, that is. Carol, our favorite nephrologist, Bakersfield. My gosh, it's always high 90s, 35C. Tom is in the Oregon Coast Range, 25.6C and dry. Rita's in southwest Idaho. Visto, 16 and damp but pleasant in Sydney. One of these days I have to get back to that part of the world. You know, I've been to Australia twice. And I had 2 trips to Australia planned last year that I had to can. So I have to get back. I have to get back to Singapore. I have to get back to Australia. I like to check out some of those other countries like Cambodia. We had a guest on TWIV, one of those TWIV episodes from Australia we had, which I couldn't make in person. He's Eric Anderson from, is it Anderson? From Cambodia, where he does lots of surveillance. And he said, you should come and look at it. Fly to Singapore and go to Cambodia, and then back to Singapore and go to Australia. Yeah. Gallbladder 8 is in Bayou Land, Baton Rouge, 27C and muggy. Had Dixon on. This reminds me of Dixon because he was from Louisiana, New Orleans. And he was on this a couple of times. We always had a good time. Remember Dixon? Too bad. Will wants a brief report of ASV. We heard about it for months, but now nothing. I can't give you a brief report because I missed 2 days, and then I was in and out of sessions because I was doing other things. So I can't. Maybe I'll think of who else was there. Maybe Angela remembers something. She went to some. She went to some sessions. I could ask her. Logotic is in Chicago, where it's 21C. John wants to know, do viruses act any different on the space stations? No, they get in cells, they reproduce, and they come out. As far as we know, they behave in the same way. I don't think people have studied it very much, right? They're, they don't want to do virus experiments in space. Maybe they could do bacteriophage stuff, but I don't know of any. Let's see. Let's take a look. Have phage experiments been done on, what do we call it, ISS, right? Yes. Phage have been studied on the ISS. They wanted to know how they did in microgravity and in radiation. In one experiment, examined the dynamics between T7 and E. coli. It worked. And they found mutations in the viral genome that improve fitness in space. That's cool. They also have looked at bacterial genomes, of course. And the idea is to look for adaptation in space. And there's some evidence for that, yeah. It's a good question. Veronika Privyet, welcome back from Moscow, 14C and rainy. Veronika was on Tweevo this afternoon with Nels LD. Leo is at the California coast, 13C, and foggy inland 2 miles. It's 25C, yeah. Melinda wants to know if the CDC has updated measles. CDC, measles update. It does it on Wednesday, right? Updated on August 20th. Yes. Yes. As of August 20th, 1,375 confirmed cases by 42 jurisdictions, 35 outbreaks in 2025. 87% of cases are outbreak associated. There you go. Vaccination status, 92% unvaccinated. Yeah, they're all there. It's all there. So you can go look at it. Just Google CDC measles. You'll get right to that page. Thank you for bringing it up. Rita, 92F. Wow. In Idaho. Daniel is in Brasilia, 23C, and dry air conditions. Julie is from North Central PA, where it is 68F. Laura's in Washington, PA, 24C and breezy. Jill is in Wilmington. Excuse me. Now, it's very hard to be sane with this dumb. Oh, I read an article on the train about Biondi, the AG. Oh my God. Horrible person. Just a horrible person. I don't like when people get so polarized. The Democrats did this, the Democrats did that. It's all their fault. It's all Biden's fault. No, it's shared, okay? So don't be so polarizing. It's what you have to do to get people to vote for you, unfortunately. But I hate when they're polarized. That's all I'm going to say about politics. Peter is in Boulder, Colorado, where skies are clear and it is 32C. Actually, I'm going to talk more about politics because when politicians mess with science, I'm going to talk about it because they shouldn't be. But it's another way to get votes. Yeah, you say where the virus came from and a certain fraction of people are going to vote for you. So stupid. Leo has just been running a chainsaw. Be careful. Boy, I would not touch a chainsaw anymore for my life. No more. No more chainsaws. Nope. Elena is in Reno, Nevada. 32C. Nice weather. Nice weather. GSD Bio Jonathan. 25C and sunny. Looking forward to hearing about nucleic acid amplification. I'm sure you'll correct me, Jonathan, when I make mistakes. Sarah, Avon Lake, Ohio. When will TWIP 263 be out? I think it's coming out Friday. Let me look. Oh, darn. When did we record it? 2 weeks ago, right? Yeah, it should be out already. I'm not good with the calendar. I really am not good with the calendar. Let me see if it's if it's been. I'm sure it's been edited. Let's just take a quick look here. Sarah, Micro TV, TWIP, episodes What did you say? 263. Yeah, it's all done. When was that recorded? It must be it should have been out already. Yes, I will get it out. I'll get it out tomorrow. It's going to be late tonight. I want to do it tonight. Okay with you, Sarah? Eliza, 19C in Southern Ontario. Everybody likes Dan Wilson coming on. That's good. Well, you asked for it. That's good. Hans from Ecuador, 28C. Nice to see you. It's lovely weather. Live stream with a lecture. You know, I taught a whole course live stream. That was fun. I forgot what year that was. I don't remember. Pete is in London. 15C at 1 a.m. Wow. And we know what autumn is. We use that phrase here in the U.S. as well as fall. Denise is in Bergen County, New Jersey. Denise, I grew up in Bergen County, Upper Saddle River. I was there from when I was 8 years old until I went to college. Many, many years. I went to grammar school and high school in Upper Saddle River, Bergen County. Oh, yeah. Michelozzo Michelizzi. Gainesville. Nice, nice, nice. His student. 31 degrees. Daniel wants cat viruses too. Yeah, we could do cat viruses with Angela as a guest. Yeah. Yep, for sure. I look like a lot of feline parents. How do I look like a feline parent? I don't have a cat. Rita gets charged up hearing about zoonotic diseases. That's what they are. They come from animals. They don't come from labs. Sorry, all of you lab leakers who think you know it all. Didn't come from a lab. It came from nature like every other friggin' human virus. Jeez. There you go. You were at the Tweevo live stream. That was good. I'll tell you about it in a moment when I do some news. Michelozzo is Michael King, University of Florida. Met Rich Condit when he was faculty at UF during internship. That's cool. That is cool. Two great live streams in one day. Thank you, Fields of Green. Really appreciate it. Melinda's cat is going to participate in that episode. This week in Catology, the podcast that makes you meow. Very good. I like that. Doug, Redondo Beach, 2 blocks in the Pacific, hot, sunny 75. I'd like that. Yeah, that would be fun. Chris L says you're supposed to like it because you look forward to it. Technically, you can always unlike it if somehow you hated it. I think that's a good idea. If you like it at the beginning, then you can attract other people as well. That's the other upside of liking it, right? John from Port Townsend, Washington, 68F. Thank you for coming, John. John says, some say cat podcasts are perfect. If you're a cat lover, yeah. Marie is in Boston. Welcome. Good to see you. Let's see. I will be in Boston in September. I'm going to the Harvard microbiology retreat, so it'll actually be outside of Boston. Michelozzo, Michael King, background in infectious diseases, infection control, microbiology, biostat, epi, med lab, clinical scientist. Welcome. I've seen your name before, though, because I remember a name like that. I love it. Kevin is in San Francisco. 75, sunny and dry. Great weather here means sweltering avian flu spreading in cattle close by. Science is not fixed. It always changes. Mushfikul. Welcome. Where are you from? How's the weather where you're from? Good to see you too. Peter says it bakes in Bakersfield. Thanks, Michelozzo. You know, I am Italian American. I'm first generation, and I love pronouncing Italian words. I don't get them all right. Some of them are very hard, but Michelozzo, Michelizzi, I love it. Airy ozone is in Asheville, North Carolina, 23C, and loud thunderstorming. You know, I am not a sports fan, but I read an article. I just read part of an article in the New Yorker about Bill Belichick being the new football coach of UNC, right? And they're paying him many millions of dollars. I can't remember. 5-year contract. I think he's getting $5 million a year. Kind of ridiculous. I mean, they don't even have a great football program. Why do they have to have this guy and pay him so much money? Anyway, that's why I'm thinking of that, because of North Carolina. Yep. Michelozzo says, Rich Condit was a gentleman I met my senior year as a University of Florida student athlete. At the time, I was an infection control practitioner working with infectious disease docs. Yes, Rich is a gentleman, always. More so than I am. I get mad. I get wound up. I get riled up. Rich is calm all the time. Okay? Visto, you should check out some Malaysian islands. Too. Lots of tropical viruses to see there. I'm not going to be seeing any viruses. I don't plan to catch them. But yes, there are lots of places to go, and hard to do it all in one place, right? Melinda said, I forgot the place. North Carolina, cloudy with a hurricane, 78F. I forgot the place. You don't remember where you are? Or you don't want to say, that's fine. Or I forgot the place is the name of it. Elena, prepping for a new semester that starts Monday. Yep. And grateful for all kinds of new stuff I learned from TWIV and get to use in my intro bio classroom. So lenacapavir is going to go into the virology course that I teach next semester, spring semester for sure. And also, we did this paper on immune yesterday. It's not going to be out for a while, but it was Kathy Spindler's pick a couple of weeks ago. They did a vaccine trial. Well, they did some preclinical work in a trial in humans of a vaccine where you put the vaccine antigen on dental floss. Actually, they did it in mice. Mice have those two front teeth, right? They have two big front teeth, and they could put the floss up and down. And there's this tissue below, between the teeth, the gingival sulcus. It's apparently rich in antigen presenting, so it's a great place to put antigen. And these mice made a great immune response. They tried it with influenza antigen, and they were protected against influenza virus severe disease. And so how to floss a mouse. It's so funny. And they bought it at CVS. They put the flat stuff so that the antigen would stay nice and even on it. And then they tried it in people. All they did was they give people those little, I don't know what they call them, a little handle with a little piece of floss. Right? You could do it with one hand for people who are having trouble with two, because they figured that would be a more controlled way to deliver it. And they just put some dye on it, and they showed the dye was delivered in the right place. So it's all very promising. And 70% of the people said they would take a flu or a COVID vaccine on Floss, whereas normally they don't want it because people don't like needles at all. And at the end of the paper, they said it's only good for people with teeth. You need to have teeth, which means if you're very old and you have dentures, you're not even very old. Just if you have dentures, that's not going to work. Or if you're a baby, a young kid doesn't have teeth yet, you can't use it. So those are two populations that you want to immunize, right? So I'm not sure that this is going to work out. Anyway, it's really interesting. It's really interesting. Maureen is in Northeast Ohio. Anyway, the Floss is going to go in my vaccine lecture as a new approach. It's interesting. 67F and drizzling rain. AM Rosa 10 writes, I know that lack of long-lived plasma cells have been implicated in lack of sterilizing immunity, but how much does incubation time play in not achieving sterilizing immunity? So it's really the activation time of the memory cells, right? So if you have a recent vaccination, let's say, so you have you have a good antibody titer in your blood and presumably also in mucosal tissues. And so that will prevent severe disease and maybe even moderate disease. You will get an infection, but very little disease. But then as the antibody levels go down, then the virus will reproduce more and you'll get more severe disease. But, the T cells are there also, and they're going to mitigate against severe disease. Now, your question, how much does the incubation time play? So that's a good question. Viruses with short incubation times challenge the memory cells, right? So we have memory B and T cells. So let's just focus on the memory B cells, which make the antibody. So you get infected, and it takes a couple of days for the memory cells to sense it and to start cranking out antibodies, to get activated and start cranking out antibodies. And by that time, the virus is replicated, so you're getting infected. That's why sterilizing immunity is very elusive. It's very elusive. So if you had a longer incubation period where it just took a long time for the virus to reproduce and so forth, then maybe you'd have a better shot at it. But now, of course, if you pre-treat people with monoclonal antibodies, that's another way to really dampen down the amount of virus and reduce shedding and reduce disease and so forth. So there you go. Do viruses even feel gravity? They don't feel anything. They don't have any nerves. They have a lot of nerve, but they don't have nerves. They don't feel anything. They don't sense. They don't even move on their they don't even move on their own. They're pushed about by Brownian motion. Other things bump into them and atoms push them around. But no, they didn't do anything. No chemotaxis like bacteria. I think bacteria may be influenced by gravity in the terms of division and so forth, but not viruses. No. Tona from Long Island, welcome. Mushfikul says, is there any relationship between E-tech and viruses? So this is the E. coli or the E-techs, right? Let me check it out. Yes, enterotoxigenic E. coli. So, do viruses, is there any relationship? So that's, there's a toxin in those bacteria. Let me see if the toxin is a plasmid or something like that. I don't know if I'm going to find it quickly enough. No. So I'm not aware of any relationship. But of course, there are bacteriophages that infect those ETECs, and they could mitigate the infection. But I'm not sure exactly. Yeah, I was thinking of Kathy Spindler to give an ASV recap. She could do that on Twiv. Yeah, that would be good. Are there any friendly viruses in our microbiome? Presumably. I mean, we have lots of viruses in us that don't make us sick. So what are they? Neutral? Are they just being carried along for the ride? Or are they beneficial? We don't know. In mice, there are beneficial viruses because we can do the experiments, right? So there's a murine norovirus that can help the mouse intestine develop properly in the absence of bacteria. And in nature, there are lots of examples of beneficial viruses, but we don't have any examples in humans because we can't do experiments, right? We just don't know. Having CDC resumed measles reporting in past 2 weeks, 19 more measles cases, total 1,375. One more state is Idaho, right? We heard that from Rita last week. MN is outside D.C. Yeah, it's icy in D.C. You're absolutely right. Nasty business, right? Just nasty. Lisa, I'm guessing virus transmission by aerosol or droplets would be very easy in the tin can of the ISS. Yeah, well, not just a tin can, but people are all in close to each other, right? You can't avoid other people. Now, I don't know that they wear face masks, right? But I don't know if they're tested before they go or anything like that. That would make sense, right? So if you're positive for flu, RSV, or COVID, you shouldn't go. I don't know. I'm not aware of that. It's a good question. Gary wants to know what's the counter to Malone's concern of distributed mRNA associated spike protein fragments. The counter is that there's no evidence that this is a thing. If mRNA, first of all, mRNA turns over in 2 weeks. Okay, so there's no long-term persistence. If mRNA is a problem, then infection is even more of a problem. So it's a counter logic that he's using. There's no known problem with spike fragments being around forever. Okay. Now, if you're worried that, let's say you're worried that spike protein is going to do something to your heart, there's a lot more spike made in infection, so you need to get vaccinated. It makes sense, doesn't it? But no issues with the vaccine, the mRNA vaccines at all. And all the vaccines are based on spike, not just mRNA vaccines. So it's nonsense. He's lying. Michelozzo, bacteriophage studies on the ISS are focused on understanding how these viruses behave in the unique environment of space, particularly microgravity. Polka Dot Pup, North Texas, 91F. Measles, yeah, West Texas declared it over with no new cases in 42 days, right? But obviously it's still going on in many other states. Yeah. Susie W, 22C and cloudy in Akron, Ohio. Welcome. John, thank you for your contribution to science communication. Maybe it will contribute to a trip overseas. Was the last visit to East Asia in 2019. Yeah, I was in Singapore in December, just as SARS-CoV-2 was ramping up, and we didn't even know it in China. It was the first week of December, and the virus was certainly circulating by then in Wuhan, having emerged from the animal market previously. Yeah, I enjoyed Singapore a lot. I did like 5 podcasts there. Boy, that was the days I was young. I carried all my stuff. I recorded myself and brought it back and edited it. I'm gonna have to record it myself again. But I did that in Montreal. It's fine. I can handle it. I'm in good shape. Yeah, 2025 is a horror movie. Yeah, you're absolutely right. Aubrey is in Richmond, Virginia, dodging hurricanes at 26C and overcast. Mushfiku says that politicians be like, you make a virus for me, operate it through my command. I mean, what do they know about viruses? Just insane. You'll see the news item I have. Yeah, they don't know anything about viruses. They have a few words that they search for. Melinda, can you do a mini lecture on viruses causing post-virus conditions or triggering chronic illness? Oh my gosh. Oh my gosh. That would be a maxi lecture. I could try. Let me write it down. I've abandoned stickies. I now use my little field notes. This is not an ad. I bought this myself. These are really cool, little notebooks with lines. You can get lines or grids. But this is very good because it's substantial and it's not going to get lost. So we have post-viral conditions or chronic illness. So last time I had a request for ultra-conserved DNA elements. That's right here. You see? So these little books are good. And they sit here on my desk. I like that. Thank you. Melinda. Martha is in Charleston, South Carolina, 27C, and a beautiful sunset. Yeah, John always guesses on TWIP. You actually don't have to be right to win. We just take everyone, unless you put a one-word guess in. No, I don't. Sorry, you're not going to get a book for one word. You have to put a couple of sentences, okay? Vanity thinks that the online course was fall 21. That may be. You took the regular course during the spring, I believe. Would you share your first meeting with Elio Schechter, who passed away recently, right? He was, oh man, I think he was almost 100, if not 100. I don't know. He was on TWIM for a couple of years. Where did I meet him? I don't know. Probably at an ASM meeting. I have a nice picture of me sitting and talking to him. At an ASM meeting. So I had him on TWIM before I even met him because someone had I don't remember who, someone had recommended. So he joined because Margaret Mafful Nye decided she had enough of TWIM and she left. So we needed someone else and someone suggested him. So I did talk to him, but I didn't meet him till later. It may have been at a TWIM at ASM also that I first met him. But he was a wonderful guy. He knew so much. He had so many stories. He was a very kind person, had a great sense of humor. And he gave me a book. Yeah. I don't know why he gave me this, but it's called Naples 44, okay? And it's a World War II diary of occupied Italy. So I think he was in Italy at this time. So he thought I should read this and get a sense for what it was like when he was there. Yeah. Richard DDS, Sunny and 79F in El Segundo, California. Why did I choose to be a virologist? Actually, virology chose me. Virology chose me. It drew me in. That's how everything works. Everything you don't make choices. They draw you, the fields draw you in. So I graduated college, and I wasn't going to do anything. My dad wanted me to be a doctor, but I applied to one medical school. I was really not interested, and I didn't get in. And I graduated, majored in biology. So I got a job. I went home to live there, and I got a job in a lab as a technician. I was doing microbiology experiments, isolating bacteria and identifying them. I really liked that. But I realized I didn't know anything. So I needed to go back to school. And in that year that I was out of college, I read a book called Fever by John Fuller, which was about discovery of Lassa virus in Nigeria. Really the first emerging virus that we discovered. Not the first one that was known, but the first one discovered. And I thought, this is just amazing that viruses can go from animals to people, non-human animals to people. So I said, I have to be a virologist. And that was it. So it drew me. That book drew me in. And so I went and did my PhD at Mount Sinai in New York. And the rest is history. I listened to people along the way. I had good mentors. Yeah. So, okay, viruses. I meant it looks like there are a lot of feline parents here. Okay, people with cats. Rita's collected a few new zoonoses. None came from a lab. Why don't we have a vaccine for scarlet fever? I don't know. Anybody know? No vaccine. Let's ask a question. Why no scarlet fever vaccine? Says that well, we have great antibiotics to treat it, and there's a diversity of strains that cause it, but I think that's surmountable. Cat viruses, car viruses, that would be interesting. Those would be computer viruses. Could it be possible that cat leukemia can be healed using a series of over 80 shots? I don't know. Maybe Angela will know. I don't look cat-like. Not at all. I don't have any kind of cat eyes. I don't have cat ears. No, I'm not cat-like. Feet made a good Putanesca. Oh, that's good. Gave it plenty of time to thicken. I like Putanesca. Melinda writes, given our overall level of trust in our politicians, it's not a wonder why people think they would participate in germ warfare, whether it's true or not. It's not that far-fetched. Pet Granny is in the Twin Cities, which 26C and sunny. What I learned today, I am a postdoc now, fortunately / unfortunately. I work in structural virology. Love your programs. Thank you very much. You'll be good. You'll be okay. You got to think positively. It's going to be all right. Jeff, happy Wednesday from Elgin, Illinois, 22C and sunny. Crazy lit as in, did COVID provide a unique opportunity for scientists to study virus-related long-term syndromes? Yes, for sure, because so many people got infected, right? And then they pretty much synchronously developed long COVID. And so suddenly doctors are like, oh, this is a thing. And the MECFS people, right, who never got much respect, now suddenly they're getting respect because, with SARS-CoV-2, yeah, you can get long COVID. So you must have had some virus infection. The problem with MECFS is that all the infections are happening asynchronously in many people all over the world at different times. So it's hard to track down. But here we had COVID infecting everyone, and I had COVID and I got long COVID. That was hard to do and remains hard to do. But CFS is probably triggered by a variety of viruses as well. Yeah. So yes, it was a unique opportunity, continues to be, because we don't know the etiology and we don't know the treatment. And that needs to be figured out because there are many millions of people suffering from it. Yeah. Carol says, we had a mild summer here, fewer triple-digit temperatures than last year. Wow. I remember driving through, actually it was Sacramento, which is probably similar. It was just the temperature went up like to over 100, and then as you got back to the coast, it went down again. Really remarkable. Pete used an Italian pasta sauce named in a post. I think I know why YouTube blanked. Oh, I don't know why it blanked out the whole message. Well, you got Putanesca in there, but they wouldn't know because it's an Italian word, right? Brian from Montreal, 19 and nice. Welcome, Brian. You could go to the name Putanesca. Well, that's what it is in Italian. You know what Putanesca is. Ay, ay, ay. Belichick, 50 million plus incentives for 5 years, and I think they also have to hire one of his kids to succeed him. Ay, ay, ay. There's a place in Colorado called No Name. Yeah, well, I would guess there would be no name in a lot of places because it's clever to do that, right? Carol got her Tdap booster in the ipsilateral arm, the same arm that you got the first one in for anticipating arrival of granddaughter in October. That's cool. Protect the granddaughter by getting vaccinated. Go Gators. I wish I had taken a class with Rich Condit, but I was in a different program. Well, you can still listen to him on Twiv. He likes to lecture. Can they do like a Listerine strip? For what? For vaccines? I don't know why you need the Listerine. Worm Talk 94. Can I come visit the incubator? Yes, of course you can. I recently started making videos. I'd love to see your setup and learn from the best. I don't know if I'm the best. I get it to work. You know, I had a lot of stops and starts in the beginning because I didn't know what I was doing. But I learned. I'm a scientist. I can learn anything. That's my philosophy. Scientists can do anything. And someone said, well, could you learn to play the piano? Well, yeah, if I had to. I don't want to particularly learn the piano. I actually would like to, but I don't have time to learn the piano. I would. I could do it. Sure, why not? I could do anything. But some things would take a lot of time to learn, so there's no point in doing them. And for example, could I learn heart surgery? Yeah, but I don't want to go through all the training to do that, okay? My dad did that. That's enough. Anyway, you can send me an email, vincent@microbe.tv, and we could set up a time, Worm Talk 94. Tona says there's an invention that the company puts peanut protein in toothpaste with the thought that the low threshold could desensitize people with peanut allergy. I don't know. Does it work? Not sure about that. Seems, as Logotic says, that seems bold, right? Because you could get more than you bargained for. Hello, Angry Penguin. Good to see you again. Could eye drops, John asked, be tried as a way to vaccinate? I don't know if it's been tried. Let's try it. Let's see. Eye drop vaccine. So it's a good idea. They actually do it in chickens, I believe, for influenza. But yeah, there's a good amount of immune tissue there. And it's been looked at for SARS-CoV-2 and influenza, for SARS-CoV-2 and other pathogens. So yes, it's been investigated. Good point, John. Krishnan from Malaysia, welcome. We were just talking about Malaysia. Someone said I should go to some of the Malaysian islands. So tell me where I should go. So I was thinking of going to Singapore and maybe Cambodia and so you should go to the Malaysian islands. So they have no way to know if they're in space or in Jupiter's gravity. Who? The viruses? No, they don't. Vanity says, I think the floss works because it causes micro trauma to the gums. That may very well be, but it also is a tissue rich in antigen presenting cells. I think that helps. It's a unique tissue. It's better than the sublingual or the buccal tissue in terms of immunizing, because those have been tried and they're not as good. Yeah, we just went through the measles numbers, Richard. It's as of August 19th. Lagodic, right? Probably as many people afraid of putting things in their eyes as there are people afraid of needles. I have a trouble putting drops in my eye. I can do it, but when the optometrist or ophthalmologist does that, my eye always closes. I try as much as I can to keep it open, but it always closes. Kang says, while discussing lung functions and looking at pictures of polio patients in iron lungs, my granddaughter turns to me and asks if she's vaccinated against polio. Smarter than RFK Jr. That's great. How old is your granddaughter? I'm just curious. Yes, virulence plasmids and ETEC. I was looking to see if the toxin was carried on a bacteriophage and delivered, but I didn't see that. But I could see plasmids, yeah. Yep. Angry Penguin, there's a Canadian political YouTuber that I had subscribed to. A couple of days ago, she did a product ad for COVID vaccine detox, so I unsubscribed. Yeah, it's the right thing to do. Don't give them any air because that's ridiculous. There's no such thing as COVID vaccine detox, and you don't need it. Yeah, it's nonsense. But it gets them attention. It gets them attention. Astronauts are quarantined for several weeks before they launch. Okay, there you go. In Chicago, there's an iron lung in the Museum of Surgical Sciences. Wow, that's cool. There's also an iron lung in Munich at the Technology Museum, which, by the way, is incredible. They got everything from telephone poles to DNA sequencers. That's a nice range, right? But yeah, they have an iron lung too. It's pretty big. Rita says another new case of measles in an unvaccinated child in Idaho. 3 so far. All the cases are in the northern part of the state. They haven't found a relationship between cases. There are likely many unreported cases. Yeah. Yep. Is nasal spray vaccination harder to have a precise dosage? No. Flu mist, you have a syringe. It has a certain volume and right? It goes in now. Could drip out, I guess. I don't know if they tell you to hold your nose for a few minutes or what, but I think it's okay. But not as good as a needle, right? And that's the thing about floss. It's not going to be the same dosing. It's going to be slightly variable, but I think it's probably okay. Yeah, so these guys refer to the Biodistribution Study where you put 600 times dose and see where the radio-labeled particles go. I don't care, right? I don't care. It doesn't happen in people. It's not an issue. There are no major safety issues with mRNA spike vaccines. They're making stuff up. Malone is just pissed off that he didn't get any credit. He actually does get credit. Perpetual Sky. What do you think about the Alpaca Nanobbody that blocks Nipah and Hendra? Could it be a treatment? Yes, I think, when you have these Nipah and Hendra outbreaks, you tend to have clusters. So if you had an index case, you could treat everyone else, like Hendra typically in racing horse farms or horse farms, and you have your first case, and then you immunize all the animals and maybe the people as well. Maybe you need to do the animals, and then it's a one world health approach to protecting people. Same with Nipah. If there's an initiation of a Nipah outbreak, you could protect people. Now, if you could make an antibody that lasts 6 months, then it's twice a year. That would be fabulous, and I think that might be doable. But yes, this is all doable for sure. But we need to do the research. And, RFK Jr. said we're going to give infectious diseases a break. Such an asinine statement to make, which would mean all this good research isn't going to be done in the U.S. Really stupid. Nevitz, for what it's worth, it's 6C atop Mount Washington, New Hampshire right now. I know the Apollo crew were quarantined before flight. A simple infection with a blocked nose could be very dangerous in a spacesuit. Yeah. Christina in Houston, Texas. Thunderstorms 97C. Yeah, it might be multiple mini lectures for your post-viral chronic diseases. Yeah. Christina recently bought both volumes of Principles of Virology. This course wasn't offered in undergraduates. Wonderful books. Thank you. Yeah, many places don't have many places don't have virology courses, but they can take mine online for free. Gary writes on the Malone distribution spike protein. The articles are online. Could scientists counter the articles as Microbe TV did the lab leak? Many scientists have, yes. And frankly, I don't think people would listen to it. If I spent an hour on Twiv debunking it, or Dan Wilson debunked it, people wouldn't listen. Yes. Toner writes, Elio and parents were fortunate to be accepted into Ecuador. I believe he said that his ship was torpedoed. YZ writes, to improve SciComm, would you support making all conference presentations of taxpayer-funded research public and free online after X days of embargo? I think that would be great. But, these conferences are a way to make money. Now, the ASM, ASM, the conference is a big source of their annual income. For ASV, it's a big source, but they also fund many, many, many travel grants to trainees. So they don't make money on their annual meeting. I mean, they make it on their membership. But they don't publish the talks online. But I think it would be great to do that. I think more people should be able to see what's going on forever, for real, right? The social media account posted a good episode of TWIM 200, I think. It featured Elio, and he describes his lengthy career at the beginning. Yes, so TWIM 200, was it? No, I think it was another number. Or maybe it was 200. Yeah, we went out there and interviewed him after an ASM meeting. This is a really good episode. Really good episode. Danny, welcome. Good to see you again. Has AI made it into your line of work? Where do you see it being used most? Well, yeah, of course it's in science already. And I think there are a couple of uses that are very good. One is to handle big data sets and do analysis. And the other is to do protein structure predictions. And those are very good at that. Spamlet, sad to hear about Elio. I used to love his stories and wide experience as much as Dixon's. I listened to all the early Twips and Twims. We are lucky you've had such greats recorded. R.I.P. I'm very lucky that all these people are doing this, and it's all recorded there forever. Yeah. Mark writes, streptococcal Strep pyogenes makes a protein that mimics cardiac tissue. Might be tricky to make a vaccine. Do viruses ever get sick? They can be interfered with, yeah. Other viruses can interfere with them and make them reproduce poorly, for sure. On Elio's website, he compiled dozens of entries about his life. Yes, it's small things considered. That's great. Any chance of a vaccine against Lyme with a Y? I'm sure it's being worked on. In fact, we talked about it recently, briefly. I looked up the trials that are being run. So, yes. Putanesca sounds a bit bawdy. Well, of course it is. It's a bawdy word, but it's the pasta was supposedly developed by a puta. Okay? So chill out. It's just a word. It helps that you love viruses. I do. I do love them. Except when they make me sick, then I'm like, hey, I'm your biggest advocate. Why are you doing this to me? A 15-year-old once asked, if you had infinite time, could anyone learn anything? And then Artemis writes, yeah, maybe, but you need to want to. And that's the key. If you don't want to learn, you're not going to learn it. Yeah. Yeah, Putanesca is exactly what you thought it means. Look up the history of pasta Putanesca, and they'll tell you that it was invented by a puta. She used to give it to her customers. Look up the story. Yeah. Oh, if you come to Singapore, I'll show you around. Thank you very much. It'll be lovely. Virus in mouth studies have shown those with poor oral health are 8 times more likely to suffer severe COVID. Interesting. Maybe those mucosal immune systems of the mouth are important. Have you heard anything more about nasal vaccines to stop transmission? No, I haven't heard, and they're not going to stop transmission. They may reduce it. They will not stop it because you're always going to get replication. Even if you have local immunity in the nasal cavity, it takes time for the memory cells to start making antibodies. And so in that time, you will have replication, you will have shedding. It will never be sterilizing. You heard it first here. Israel, are phages being more used along antibiotics in the U.S. now? No, very few. They're just isolated incidences of phage therapy being done in the U.S. There are other countries where there's a little bit more, but they're not yet being used routinely. No. My dentist says flossing disrupts the bacterial film on the teeth. Right. It's meant to take out the plaque, right? Because if you don't floss, it will build up in between your teeth, and that causes a problem. Then it gets down to your gums, and it causes gum inflammation. Yeah. Anybody watch the Vaccine Integrity Project yesterday and their expert presentation on the latest publicly available data on flu, COVID, and RSV vaccines? I did not. True experts refuting the faux ACIP. Wow. Oh, she's 8. That's lovely. I just think that's great. Okay. Do antiviral drugs cause dysbiosis? I know that the triple therapy, HIV triple therapy causes all kinds of side effects over the long term, but acyclovir disrupts the gut microbiome. So yes, some of them do. Okay, I am going to stop here and just do some news and then a mini lecture, and then we'll get back to questions. So I have a news article. I have to take this off. I have a news article from Science. Let me share it here. I don't know if I can get it. Oh, yeah, here we go. Let me share the screens. Not now. Stop doing all these notifications. NIH, this is from July 11th. This is a while ago, but there's a reason I'm saying it. By John Cohen and Jocelyn Kaiser. NIH suspends dozens of pathogen studies over gain of function concerns. So basically, the NIH, in response to the White House, has begun a crackdown on dozens of studies it was funding. So this is oversight of gain of function research, potentially dangerous. And so NIH has identified 40 projects that may meet the definition of dangerous gain of function. Erring on the side of caution, all projects potentially meeting the definition are being suspended. Okay, this is U.S. universities, NIH. And the reason I am, and this is nonsense, right? This is just nonsense. The reason I am bringing this up is because one of Nels Eldy's grants was flagged as potentially dangerous, and you can't do these experiments. What did they flag? He's taking mouse hepatitis virus, it's a coronavirus of mice, which has never been known to infect humans, and passing it in human macrophages in culture. Okay, the reason to do that is to look at how a particular gene is evolving in macrophages, which have immune defenses against the coronavirus. So they flagged this because it's passing an animal virus in a human cell, and so that's potentially dangerous and could cause a pandemic and kill a lot of people. This is just nonsense. This is nonsense. I can't say it's not going to happen, but you use the correct containment which is commensurate for what you're worried about. And this caused Nels a lot of upset, and he had to go through hoops to deal with this. In fact, the work has already been published, and we talked about that on Tweevo today. It's lovely work, and no virus arose that infects people. But what I wanted to point out, so they're saying we're suspending these experiments over an abundance of caution. Nobody ever had to suspend any such experiment like this over an abundance of caution because there's nothing to worry about. But let me just tell you what two individuals have said about this. I have some quotes. So one person said, ill-considered self-censorship by PIs, principal investigators, and the effects of intimidation on PIs might be some of the most damaging and both figuratively and literally sickening effects of the current circumstances. This is from David Relman at Stanford, a microbiologist who has railed against certain kinds of research activities. He said they shouldn't be done. Now he's saying by stopping this research, it's going to be dangerous. You are partly responsible for this current climate. What are you doing? Another quote by another person. Only a handful plausibly meet the standard. He's talking about the experiments that were shut down by the NIH. Only a handful plausibly meet the standard and in the executive order that they could result in significant societal consequences. While there are a few studies where I would want to learn more about the risks and benefits, I would predict that ending this list of studies as a whole will be detrimental to health security and public health. You know who that was? That was Mark Lipsitch from Harvard, another guy who rails against certain virology experiments. He and Relman and others have created this climate that infected the White House to say we're not doing certain kinds of gain of function experiments anymore. And now they're saying this is bad to restrict these experiments. I am just blown away by the paradox here. I'm just blown away, right? So that's why I wanted to tell you about that. Okay. Let's do a little mini lecture. We're going to talk about nucleic acid amplification methods. Let's see if we can make this a little bigger. Move this up here and down here. Okay, nucleic acid amplification. Here's what we're going to talk about. The reason I'm calling it this and not PCR is because it's more than PCR. Polymerase chain reaction, right, is the very first amplification method we're going to talk about. And it's a thermal method. You have to change the temperature during the reaction, as you'll see. And then we'll talk about derivatives of PCR like RT-PCR, reverse transcriptase PCR, quantitative PCR, microfluidic PCR. These are all thermal methods where you have to change the temperature. Then we're going to talk about isothermal methods, which means you don't have to change the temperature during the reaction, during the amplification, which is nice because, the changing of the temperature requires a pretty big instrument. So if you don't have to change the temperature, you could do it in the field in a thermos, for example. So we have loop-mediated isothermal amplification, or LAMP, and rolling circle amplification. And we'll also talk about ways to measure the product. So we're amplifying nucleic acid, and we want a readout. We want to know when there's nucleic acid that's been amplified. How do you do that? And this has been another evolution in itself. So it all starts with, actually it starts before PCR. It starts in 1983 when Kary Mullis realizes that just by modifying the Sanger sequencing method, you can double your amount of DNA. So what am I talking about? We talked about Sanger sequencing last time. We have a template. We have a DNA, and we have a primer that is going to prime DNA synthesis on this template together with DNA polymerase. And we use dideoxynucleotides, all four, that terminate the polymerase at a specific base. And so you can separate the fragments by size, and you can read the sequence based on what dideoxy has been used to terminate the reaction. So you do 4 reactions. This is using double-stranded DNA, which you denature, and then you use a primer that hybridizes to one strand, and you make the complementary strand. And Kary realizes that if you copied both strands, you'd get twice the amount of DNA. So you have one strand with a primer, and then you'd need a second primer for the other strand, which isn't shown here. You get twice the amount of DNA. So he did these experiments to show this. And the problem is, to denature the initial DNA, and then the product after each amplification is going to be double-stranded. So you're going to start with 2 single strands. You have to heat the mixture to 100 degrees Celsius to get that denature. And then you have to cool it and add the polymerase, and now you have 2 double strands. But to denature it again, to amplify it again, you have to heat it, and that kills the enzyme. So you have to keep adding more and more enzyme. But you do get amplification. And that was the genesis of polymerase chain reaction, that you could start with a double-stranded DNA and make 2 and then 4 and then 8 and then 16 and 32 and get huge amounts of DNA from very little to start with. So that's the genesis, that the Sanger sequencing reaction gave him this idea that you could double the amount of DNA that you made just by using both strands of the DNA. Okay? But this limitation, it was lethal because the denaturation, as I said, kills the enzyme, and then you have to keep adding more, and that's just not good. So what happened to fix it? Well, first of all, here is, where is it? Here is the reaction, basically, that we are going to set up. So in the Sanger sequencing, you take double-stranded DNA and you denature it at 100 degrees, and then you have a primer that will bind to both the plus and the minus strand of the DNA. Then you add your polymerase. You cool the mixture down to 37 degrees. You add your polymerase, And now you have two double-stranded copies of the original single double strand. And then you could denature those and get both strands. In each case, now you have 4 single strands, and add new polymerase and primers and so forth, and you would get 4 double-stranded copies. You can see you go from 1 to 2 to 4 to 8, 16. And a single molecule can be amplified between 1 million and 10 quadrillion times. So you can have vanishingly small amounts of DNA and amplify it to the point where you can detect it very easily. So this is the principle of Mullis's early PCR based on the Sanger sequencing. So what changed this was Tom Brock, a microbiologist at the University of Wisconsin in Madison, who was interested in what grew in the hot springs at Yellowstone Park. And he, in the 1960s, discovered that there were hyperthermophilic bacteria there. These are bacteria that can handle the heat. They grow in the heat. And he isolated one, and he called it Thermus aquaticus. And he studied it for many years, and he found that the DNA polymerase, of course, is stable at 100 degrees because it's living in water that's very hot. And 20 years later, Kary Mullis and colleagues say, wait, this enzyme, this DNA polymerase from this Thermus aquaticus, not aquatics, it should be aquaticus, can withstand the multiple cycles of denaturation that we need to separate the product DNAs, right? You need to heat it after every polymerization. So you make 2 double-stranded molecules from one, but to make more, you need to denature these. You have to raise the temperature. And now this enzyme that can stand 100 degrees is perfect because you don't have to keep adding it. So this enables automated PCR to happen. A machine can do the whole reaction, and you don't have to add enzyme after every cycle. And that, in my opinion, revolutionized this technique, because otherwise it would never have taken off, I think, having to add fresh enzyme. Or maybe someone would have figured out how to engineer an enzyme to make it thermostable. But Tom Brock was just curious about what grew in these hot springs. He didn't say, I'm going to discover a thermophilic bacteria whose polymerase is heat stable and which will enable PCR in 20 years. Of course not. It's not how science works. You just are curious, you follow your curiosity, and good things happen. And people don't get this. They say, oh, you have to cure diabetes. Okay, you can try to do that, but you also need to let scientists follow their curiosity, okay? So now we have polymerase chain reaction, and if you want, you can amplify DNA. But if you want to amplify RNA, it's no problem because we have an enzyme reverse transcriptase, which will copy the RNA into a DNA. So you do reverse transcriptase PCR, RT-PCR, and you can quantify RNA like in RNA viruses or mRNAs in cells. Okay? So this is automated using these machines, which we had 4 of in my lab, and many labs have dozens of them. It's essentially a heat block in there, a metal block that's very good at holding temp. And you put small tubes in it, and then in the tubes you have assembled your reaction mixtures, and they're closed. You close the lid, and then you program it to do, you can change the temperatures, you can change the annealing temperatures of the oligos depending on their sequence, and you can change the extension time of the polymerase, and you can fiddle with the number of cycles that you need to do of amplification to detect your product. Okay, and these are great to do that in, but of course they're heavy and they need electricity, and you can't bring them out in a field. It's not very practical. You can, people have done it, right, with generators and so forth. But as you'll see, there are better solutions to that. So here's how it works. So this is one cycle of polymerase chain reaction. First, you heat the DNA to separate the strands. This is typically 94 to 98 for 20 to 30 seconds. So the machine is programmed to do this. Then you lower the temperature to allow the primers to bind to the target DNA, because at 100, they're not going to bind. They're going to remain separated. So this is typically 50 to 65 for 20 to 40 seconds. And again, it depends on the sequence of the primer, because the sequence of the primer will determine the annealing temperature. Then you raise the temperature to the temperature that works for DNA polymerase to make new strands. And this is typically 72 degrees for 1 or 1.5 minutes per kb of target. Now, most people are doing very short amplifications because they just want to know if something is there or not. So it's a quick reaction. And that's it. Denaturation, hybridization or annealing, and polymerase extension. So you repeat this multiple times, and you usually don't go over 35 or 45 cycles, and you amplify the target DNA a huge amount. And you can use it to clone genes, you can use it for detection, and so forth. We'll talk about that later. Then we have, after so many years, so we want, if you want to know how much DNA you are making, the process I just told you is not quantitative. It's an all or none. It will tell you, yeah, there's a DNA of that particular sequence in the reaction mix, but I can't tell you how much of that DNA was originally there. So quantitative, or qPCR, was developed to measure DNA produced during amplification. And you can do qPCR for DNA, or you can do RT, reverse transcriptase qPCR. All right, so this is quantifying the DNA made during amplification, and you can use this to determine the copy number of viral RNAs or DNA, right? And you say, oh, this patient has X copies of SARS-CoV-2 RNA in their saliva or something like that. And that's based on quantitative PCR. You can do gene expression. You can say how much mRNA is being made. You can even look at genetic variation, single nucleotide polymorphisms, by making primers that match or do not match a site that you think is mutated. We'll take a look at that later. So how do you measure the DNA that's made? This is done by using DNTPs, deoxynucleoside triphosphates, that are labeled with a fluorescent tag, for example, or you can use a dye that binds to double-stranded DNA. So as DNA is made, you get more fluorescence, or you get more of this dye. It's called cyber green, that binds double-stranded DNA. And then you can quantify the DNA made by measuring fluorescence or cyber green. So this then allows you to quantify, and a key aspect of quantification is the cycle threshold, or CT, which you have heard. If you listen to any of our podcasts during COVID, you heard about cycle threshold. What is CT? It's the number of amplification cycles. Remember, denaturation, hybridization, polymerization, that's a cycle that you need before you can detect the DNA. All right, and here's a curve. Here are two curves to illustrate this. On the left is we're measuring fluorescence on the y-axis, right, and the number of cycles, and the sample is in blue. And you see we're measuring the fluorescence of the sample, and it's going up. And eventually it plateaus as you run out of reagents. The CT value is when this fluorescence exceeds the background level, or what we call the threshold, above which you're confident that the signal is not just fluorescence that's generated in the system, which happens, a background fluorescence, right? And you can see that in this graph on the left, the CT is exactly where the reaction exceeds the threshold, but on the one on the right, you can see the CT value is slightly higher than that. So the CT value is important because now you can compare different PCR reactions. And so on the right, you see we've done amplifications with a lot of DNA, high copy number, or a low copy DNA, right? So when you don't have a lot of DNA on the right here, it takes more cycles before you reach the threshold, before you reach the CT. And so it's the inverse of what you would think. You have a low copy number, you have a high CT value. You have high copy number, you have a low CT value because you don't need so many cycles to get up to the threshold. Again, the CT is the number of cycles you need to reach that threshold. And you can go from 0 to 40, say, so it could be any number in between. And so this is a very quick and easy way to do quantitative PCR. If you really need to be precise, you could use a known quantity of DNA as a comparator, right? And then you can say, I put in X nanograms of DNA, and so this, and you can extrapolate for your sample. But if you want to compare different samples, let's say you have two people who you've done a nasal swab, and you want to know if they're infected with SARS-CoV-2. If you want to get some idea of how much viral RNA they are producing, you have to either put the exact same amount of material in, but you can't because the swabs are not accurate, or you need to use an internal control. So you can take mitochondrial DNA, which is present in everyone's nasal sample, and you could amplify a gene from there and then normalize the CT for the SARS-CoV-2 to that. So, early in COVID, we never actually did cycle thresholds. We just said, oh, you have a positive PCR, and therefore you need to stay home for 2 weeks. But, the PCR could be detecting a lot of DNA or a small amount of DNA, and maybe you're not infectious. And that's always been the problem with COVID diagnosis. Then we have microfluidic. We'll get back to some of these issues in a minute. Then there's microfluidic PCR. Basically, they're very small tubes that can route all of these reagents and do the reactions here on a slide, for example, a slide-sized apparatus. You could add the sample at one end, and the lysis could occur right on the slide, and then these small tubes will mix the sample. You add your PCR reagent, and it's mixed again. Then PCR happens. You can add dyes in and mix again, and then the output. So it's just essentially doing all of these reactions in small tubes, and you can multiplex this and do a lot at once. So that's just microfluidic. A nice example of how to use PCR to detect mutations in your DNA. And if you go to 23andMe, this is basically what they do. They do PCR, SNP PCR, single nucleotide variations. So there's a gene for earwax, and there's a single base change that gives you a phenotype, and you can design primers that will be sensitive to that change. So in one method, for example, the primers are made to be specific to either the variant or the wild type allele of a SNP. So here you have two examples where we have a primer. Here's the short primer here. And you've made it so that the 3' end is now matched to say the wild type sequence. And then you have another one where if you amplify this with the wild type sequence with a DNA that has a SNP, it's going to be mismatched, and so the PCR won't work as well. So you could make wild type primers, or you could make SNP primers, and typically you use both. And that's how you can tell. And this is used for all kinds of SNPs, not just in genetics, but also in cancer biology. If you want to see what kind of cancer driver you have, you can look by SNP. Okay, so those are the thermal methods that we've talked about. Let's talk about some isothermal methods. Now, this is lamp loop mediated isothermal amplification. This is like PCR, but it's done at a constant temperature. So like means it's amplifying the DNA, but one temperature. So that's great because you don't need a large apparatus to be raising the temperature and then cooling it down, right, quickly. So essentially what you do is you use a DNA polymerase, what we call a strand displacing polymerase, and 6 primers. So here, for example, is our template. And we use a primer here to make this product, and then we use a second primer to make the second product. And what these primers are doing is they're adding loops to the end of the DNA, right? Because the DNA target is right here in the middle, and then the priming is adding the loops. And then you use primers to the loop, so you don't have to have primers to your actual gene. You use primers to the loop to amplify this sequence. And, you use multiple primers at the same time, and the DNA polymerase goes around the whole structure and amplifies it. And it's one temperature because you don't need to have any denaturation. The polymerase would just keep cranking out copies of this template without having to switch temperature. So this is often used, and this was developed for COVID tests as well, and many other applications. Then we have rolling circle amplification, which is another isothermal method. Again, one temperature. So again, you have a target DNA, and you make a probe, which is called a padlock probe, which hybridizes to a target sequence. You ligate this circular template, and now you take that complex and you add a primer to it, and you add a polymerase, and the polymerase goes around and around and around that circle, and makes very long concatemers of amplified DNA, which then can be easily detected. That's why it's called rolling circle amplification. And this is a strand displacing polymerase, which means when the polymerase has gone around the whole circle, right, it's gone around the whole circle, it will displace the product it already made and keep going around and around and around and around. And these are the ways that small circular DNA viruses reproduce. And so some of these polymerases are from those viruses. There can be different readouts. So we've talked about the original PCR. You could run your product on a gel and look at it and see, oh, I have a product. Therefore, there's the DNA sequence I'm interested in my original sample. And then it got to be fluorescent or cyber green for quantitative PCR. But methods have been developed where during the PCR, as the DNA is being amplified, you get a color change in the tube. So this is colorimetric PCR readouts. So for example, you can just look at it. So the fluorescence and the cyber green, you need a detector, a machine to detect that. This you can look at in the field and say, it's positive. You can use a change in pH. So the medium acidifies after DNA replication. This compound, hydroxynaphthol blue, goes from violet to blue as a result of DNA synthesis, which consumes magnesium. And so the reduced magnesium causes the color change. And then this is a cyber green, intercalating dyes as like cyber green, which can be used as fluorescent indicators. These can also be seen by the naked eye. So these work in the field so that you can now go out and do LAMP in the field with these visual indicators and get a PCR result without any heavy equipment. So PCR is not the same as infectious virus, okay? And we've been talking about this for years, and it's all based on this experiment out of the CDC years ago when Zika virus was a problem, 2015. It still is a problem, but when we first learned it was a problem. They would infect male mice with Zika virus, and the virus is shed in seminal fluids. And they measure infectious virus here in this time course, and you can see there's a peak, and after 25 days or so, there's no detectable infectious virus. But if you do PCR, RT-PCR on seminal fluids, you see that viral RNA persists much longer than infectious virus. So that's because PCR is just amplifying small bits of nucleic acid, which are not necessarily infectious. And for many RNA viruses, RNA can be found long after the disappearance of infectious virus. So early in the pandemic, people were equating PCR positivity with being infectious, and that's just not right. Why? Well, let's take a look at that. Here is a curve for SARS-CoV-2. We're looking at days after infection, and the quantity of RNA in green by PCR. And then in blue is the infectious virus determined by a plaque assay. So these are clinical samples from people, and you can measure RNA, which you can see goes out to 22 days, but the infectious virus is gone by 12 days or so, at least in this experiment. So just because you're PCR positive does not mean that you are shedding infectious virus. It could be small pieces of RNA that are just persisting. Now, if you do this in a different way, this is an experiment that was published some time ago, where you take nasopharyngeal samples from patients, you do PCR value, and then you also do culture positivity. What that means is you take a little bit of the nasal wash, you do a PCR reaction, you calculate a CT value, that's on the x-axis, and then you just throw a little bit of that nasal wash into cells in culture, and you see if it infects the cells. You just visually inspect them, and you say, there was virus in that original sample. But you don't know how much virus was present. You just know it's culture positive. So remember, low CT value means a lot of DNA is present, or a lot of viral RNA. And here, CT value of 20, they had 3 out of 4 culture positives and a very big error bar. So this is not very useful. But you can see a little bit higher CT value. 38 of 44 samples are culture positive. So having a lot of DNA as represented by a low CT is consistent with culture positivity. And as the CT value goes up, you can see the culture positivity goes down to when you get to 35 or 40, only 5 out of 60 of those samples were culture positive. But the caveat is we don't know how much infectious virus is present there. We just know it's culture positive. It may not be enough to infect anyone. And this is a real problem because doing plaque assays is not really suited for clinical applications. There has to be a way to automate them. But you would say that the chance that you're going to be, if you have a CT value of 37, it's highly unlikely that you are transmitting SARS-CoV-2. But these are the issues that are involved with thinking about that. So what do you use PCR for? You diagnose diseases, either infectious diseases or genetic diseases. You can do forensics. You can clone genes. You can amplify up DNA and put it in a vector. You can measure mRNA expression. You can do mutagenesis. You can use primers that have altered sequences, so you can introduce mutations into DNA. We should do that a lot. And you can do DNA sequencing. For many of the DNA sequencing methods that we talked about last time, we amplify first using PCR. And I think that's it. Let me hope that was useful. And if you have any questions, put them up. We have about 20 minutes left. And let's get back to the question. Hat writes, I used to laugh at the movie of Pasteur when he said all disease may be caused by microbes. Recent TWIV's virus's effect on cancer make me laugh at myself now. Oh, it's all tied in. It's all tied in, that's for sure. Why did the microneedle patch studies not go anywhere? Well, they didn't work very well. So the one study I'm very familiar with is the polio virus on a microneedle patch, and it didn't provide a comparable antibody production as virus immunogen does. And I think that's the case for other viruses as well. And yeah, it sounded great. It hasn't worked out. Tona writes, how does skin TB test work? So you're basically injecting tuberculin antigen, and if you've been previously infected, or if you are currently infected, you will get an immune reaction. So you get a swelling at the site. You have T cells coming in and cytokines being produced and inflammation happening. So you get a swelling and an induration, as we call it. I am rereading Jonathan Howard's We Want Them Infected. Most of the scoundrels he writes about are now overseeing U.S. health. Isn't that horrible? Yep. Christina writes, while grocery shopping, I saw a young father carrying a child with SLAP's fifth disease. Please tell me why. Why he was carrying the child? Well, it's caused by a parvovirus, right? A single-stranded DNA-containing virus, which can also cause disease in cats and dogs. And I don't know why he was carrying the child around. It is a pretty mild infection, but you know, it is spread by respiratory routes. So if the child is shedding, it would be shedding in a danger to other people, especially people with compromised immune systems. But that, you know, it's not something that people really think about. Man wants to know how virologists use mathematics to model how viruses behave and spread. Well, yes, this is the purview of epidemiologists. And if you want, we could get an epidemiologist on here to explain it. But yes, statistically, modeling is used to understand and try to predict transmission of viruses in populations, right? Because virologists study reproduction in cells in culture or in animal models, but the epidemiologists want to know what happens in a population. And they want to know, for example, what's the rate of spread, what can you anticipate, and so forth. And they often use statistical procedures, but I don't know. You know, it varies on a paper-by-paper basis. But often Bayesian statistics are involved in that as well. But we can get someone on to talk about that. Plaque assay is a good example of math. That's a very simple math, right? You count the plaques and you multiply by a dilution factor and you get the virus titer in PFU per mil. But, you know, predicting where an outbreak is going to go, it requires statistics and use some data on what you know, but it does require statistical procedures, which is not really needed for a plaque assay. How is the likes? Yeah, we're pretty good. 140, 122 likes. We could get a few more people liking, so that would be great. Lori listened to Immune Booster with Shane Crotty talk about an amazing job done by scientists during the first year of the pandemic. Yeah, we had him on during the first year twice, I think, talking about what was going on. Yeah. If you get exposed to two viruses simultaneously, do they fight to infect you? They can. If they're similar viruses, right? They can compete. And they have ways of inhibiting each other. But different viruses could do the same thing. But we are infected with many different viruses at any given time, right? So yeah, it's a very competitive world out there, but viruses can coexist. There are a lot of different cell types to infect. So all the viruses infecting us, they can each pick a different cell type. But if they're going to infect the same cell type, then yes, there's going to be competition, or as you say, they're going to fight to infect you. Why is there no Putanesca? I don't know. No idea. No, not sure. I have to look up the story. I don't know why there isn't that one. Canada has more measles than the U.S. That's right. We need, Tona writes, a top 10 gain of function that has led to disease breakthroughs. Can you put together a top 10 list? I could easily put together a top 10 list. But nobody would care because they would say, oh, that's all good, but how do you know one isn't going to go bad at some point, right? For example, we in my lab made transgenic mice susceptible to poliovirus. That's a gain of function for the mouse. They're extremely useful for studying the disease and also for testing vaccines, polio vaccines. So that's a great example of a beneficial gain of function, which nobody knows about. The last person on an iron lung died a few months ago. I hope it doesn't become a thing again. No more iron lungs. We'd use other means of helping the patients breathe, which would be, intubation, which is unfortunately not as gentle as an iron lung. Artemis, early 80s, my biochemistry prof was telling us that some researchers had this science fiction idea that one day we'll be able to multiply DNA in the test tube. Well, there you go. Maybe they were talking about PCR or nucleic acid amplification. PCR question, if you don't know the DNA sequence, how do you determine the primer sequence? Well, you can attach a known sequence to either end of the DNA, right? That's one approach, and then use a primer to that known sequence. Or you can use what are called random hexamers, a mixture of 6 base primers of all different sequences, of which some are likely to attach to your template and prime it, and that works very well. How does Yellowstone bacteria keep its 2 DNA strands together at such temperature? Well, they probably have compounds that enable the DNA not to denature, right? Let's look it up. Yeah, they have proteins that keep them together. They also have a high GC content. GC base pairs are more thermostable than AT base pairs, so they have a high GC content. So that's it. Basically, specialized proteins and high GC content. Good question. And we also got that from Visto. The beauty of curiosity. That's right. It's great. Similar story with GLP-1. It all started with basic research addressing basic biological questions. No one had weight loss, etc., in mind at the beginning of the research. Basic research often expands the scientific toolbox, where often the new tool gets put on the shelf until someone comes along and says, hey, we can use this to help our project. That's right. And CRISPR-Cas, right? They were discovered as bacterial immune systems and then repurposed to do gene editing. It's not what they do naturally, right? They're a defense mechanism. Yes, rolling circle DNA amplification is the process that some viruses do. So we have the herpes viruses, the genome is linear, but when it gets into the nucleus, it's circularized and then replicates by rolling circle amplification, essentially. Yeah. Should we get a COVID shot? Ask your doctor. Ask Daniel Griffin. CDC recommendations are reasonable. That's what Daniel recommends. If you are, over 65 or have more comorbidities, you should get an annual shot. That's what CDC says. Of course, now if you're under 65 or pregnant, you can't get it unless you have a comorbidity under 65, whereas pregnancy is a comorbidity. So that's just dumb. That's just dumb. But yes, if you're older, you should definitely follow those recommendations. That's what Daniel says. How do you decide what method you use? Well, for me, in our lab, we just wanted to make more DNA so we could clone it or sequence it. So we just used PCR or RT-PCR. But, the lamp and the circular methods are good for field operations. Or if you don't have a fancy thermocycler, you can just do it at one temperature. So it depends on circumstances. Important point, the primer becomes part of the DNA. That's right. So you can add restriction sites. You can add mutations. It's really very flexible. Very, very flexible. Jessica made cDNA from RNA for the first time, and we'll be doing PCR this week. That's the right term, too. It's cDNA is that first strand of DNA made from RNA. Yes, using primers that don't match is a way to introduce mutations. Fifth disease, Helen writes, may not be mild in adults. It could be immunosuppressed. That's where it's worse in, yeah. SLAPS is dangerous for pregnant women. Hello, Abdur. Welcome. Yeah, we're almost done, but you can go back and see it. The primer only needs to anneal to the template strand at the 3' end, and 10 to 12 bases is usually enough. That's right. That's why if you're looking for SNPs, you can make that 3' base the mismatch, and it'll be very sensitive to that. Where else can you get answers to microbiology questions, explanations for lab techniques and equipment, epidemiology, bad science debunk? Here and on all our other podcasts. I'm glad you like it. Thank you. So RT-PCR, reverse transcription PCR, you start with RNA and you make a DNA copy, and then you do conventional PCR. Real time, you can start with RNA or DNA, right? So it could be qPCR or RT-qPCR. And so, we use Q quantitative because RT is reserved for reverse transcriptase. And RT is also room temperature, so there's a lot of mix-up here. Should someone over 65 get the COVID shots? That's the recommendation, and that's what Daniel Griffin says. You can get a couple of months of solid protection by getting a booster for the fall COVID season. So your dad is 101. He would be recommended to have an annual booster shot, yes, not by me, but by the CDC and Daniel Griffin, who is an infectious disease physician. Because I don't do, I don't give you medical advice. I can give you polymerase advice, though. How many generations away do you think we are such that cavities will be a thing of the past? I bet a generation, and we can do this. Fluoride helps a lot too. It really does. How does the polymerase work? So the DNA is threading through the polymerase, and the DNT, there's an active site where the bases are added, and all 4 bases are tried. So let's say there's an A in the active site of the polymerase. The polymerase adds all 4 bases sequentially, and the right one will cause a conformational change in the polymerase that then allows that base to be added properly, right? So it's just like a trial and error thing. And sometimes it makes a mistake, and, that's how mutations occur. Gabriel, I finally made it to my PhD program in the U.S. from the U.K. Landed 2 days ago. Good luck. I think you'll be okay, but good luck, Gabriel. Good to see you. Yes. My sister's home with COVID. Paxlovid wasn't covered. A lot of yeah, $1,800. A lot of people can't have that. How expensive are the different forms? For labs, small labs, it's expensive. The reagents and the machine is expensive, but if you do a scale up, it can be $1 a test, right? Yeah. What about PAXS, that access program? I don't know what's going on with that. Yeah, the CRISPR, the Dannon yogurt people, it was a yogurt company in Lithuania. I don't know if it was Dannon, but they would fail, and they looked into it, and they found the CRISPR system that way. Yeah. If I were back in the lab again, what would I be working on? It's a good question. I haven't thought about it, you know. I'd like to work on poliovirus, but they won't let us because there are lots of questions still ongoing with poliovirus. What else would I do? It has to be some virus, some aspect of evolution of viruses, because I think that's something that we can get at very, very readily these days. Yeah. And wait for vaccines against bacteria that cause tooth issues. Yes, I think that would be fun. Fun. What am I saying? That would be great to have that, and maybe some other approaches as well. All right, your questions are finished. So in that case, we're finished 3 minutes early. I want to thank our moderators tonight. Who do we have? We had Vanity Nutrition. We had Les Fabi. We had Tom Steinberg. And thank you for coming and moderating. Really appreciate it. I also thank everyone for coming as well. We had 150 of you at one point, which is a great number for this program. We will be back next week. Let's see. What's the date? August is almost over, folks. It's going to be August 27th, 8 p.m. Eastern Time. Bring your questions, comments. I don't know if I'll have a guest by then, but if not, it will be me. Our questions are infinite. Yes, you can keep asking them. We could do have a couple of mini lecture topics here. So no problem ever having something to talk about. This is Vanity Nutrition's favorite community. That's very kind of you. Thank you so much, everybody. Yeah, this makes a very nice community here. We've built this community over the years, right? And I'm happy to see everyone coming back, and I'm also happy to see to see new people. Putanesca for dinner. Go for it. Not a hard recipe. Thanks, everybody, for being here. Love to see you. Till next week, please be safe and also be curious. Bye-bye.